Very good morning to all of you. If you see the program what is given to you, today's lecture is supposed to be by Annapurna. With the permission of all of you, we are shifting that lecture to another day because madam is slightly busy in the last few days for RAC. I must thank Dr. Nirmal, who is sitting here. He readily agreed to deliver the lecture today. And logically, his lecture should have been a little later because he is going to talk something on how to visualize the genomics data in a manner anybody can understand what we have done with the sequence data. So basically what he's going to speak, molecular phylogeny and some of the basic principles about the tools and techniques which is used to visualize the genomics data. Some of you may be knowing Dr. Nirmal. He's one of the excellent teachers in the division of plant pathology. He joined ICR system early 2000, if I am correct? 2003, yeah. Initially, he was posted in a small institute that was in West Bengal, that is a Duke Institute. And later on, he moved to National Bureau of National Bureau of Plant Genetic Resources, NPPGR in Delhi, as a senior scientist. From there, he moved into IRA. He moved into IRA as a senior scientist from NPPGR. And presently, he is serving this division as a principal scientist. And he has got numerous publications within the specialty of his own subject, that is plant virology, and published a lot of papers in reputed national and international journals. And he was very actively associated in different national and international societies and academies. And Dr. Anirban has really gained a good reputation among the peers and among the students as a good teacher and a good orator of the subject concerned. And presently, he is doing a lot of research activities in plant virology. And just for the information of the house, just a few months ago, he returned from Washington State University, where he was trained on genomics. So the modern subject, which everybody is aware now. I'm sure you all will enjoy his lecture. And he is not new to many of us, especially the students of plant pathology, but the rest of the students who is from different discipline, they will enjoy his lecture. Dr. Retrieval.
So, very good morning. Loud. If you don't have the energy, how I'll get your energy should infuse within me. So, well, actually, the talk was scheduled to start at 10, but it was almost 20 minutes late. Forget. Because just think about billions years ago, when there was no concept of time. The time starts from a big event, which is called the Big Bang. From there only the concept of time came. So 20 minutes is very little if you see that billions years. So that Big Bang, when it was there, then what happened during that very active environment that time? Different molecules, different things joined together to form a very basic biological molecule of the life, which is called a small RNA molecule. That RNA molecule is very catalytically active molecule, and they started replicating. And in addition with other different molecules, they started producing proteins and other things, and ultimately they gives rise to generation of this RNA molecule, and then a very small primitive molecule or primitive organism, that is the prokaryotics or primitive prokaryotes. So from these primitive prokaryotes, then all the life forms created in the earth. So whether if you talk on the plant or animal or everything started from that small RNA molecule. So we are actually brother of, or we are ancestor of that small RNA molecule. So we should have commonality, similarity between all of us. And among all this biological entity, we the human being are the most intelligent, civilized creature of the Mother Nature, and we are now trying to compete with Mother Nature. Mother Nature tried to take billions of years to evolve us, and we are trying to crack the mystery of this mother nature by just sitting on a computer to analyze what mother nature has done. So, after that thing, when human civilization develops from the river valley, we are now entering into the Silicon Valley. So, when we started our civilization, our quest for life started. We started to know what is life, how the life evolved, how all the living being in the earth is related, and what is the element which carries the information and passed to the next generation. When this quest started beginning, the science subject started beginning. The early days of the science, when Charles Darwin and Mendel, they started to analyze these things, they started to answer these questions, and then the principles of heredity, theory of evolution came, and this is the era of classical genetics. Then several discoveries made. Messier first identified nucleic acid, composition of nucleic acid was identified. It was known that DNA is the genetic material which carries all the information. Extracrystallography of DNA was made, DNA structure was identified, and we came to know that in the DNA there are triplet codon which actually carries the information and translates it into protein. So, in this era, that is the era of molecular genetics, what actually is happening, we are trying to understand how DNA is transcribed to RNA, that passes the information to the RNA. From the RNA, how the information passes to protein through the process of translation. So, you know all those things, just I am trying to put those things so that you can have that understanding about whatever the subject we are talking today. So, this is the central dogma of life. So, in this era, in the era of molecular genetics, what is actually happening? The classical genetics, the subject, the molecular genetics, and the cell biology, these three different branches of science, they are trying to merge together to give rise to the birth of another era, the era of cellular molecular biology. And during that era, along with this subject, the tremendous technological advancement started beginning. The sequencing of the DNA was done, polymerase chain reaction was discovered, cloning and recombination DNA technology was discovered. So, with the advancement of this technology, now what happens? The classical genetics, molecular genetics, cell biology, and the technological advancement, they merge together to give rise another era, the era of biotechnology. So, this era of biotechnology got tremendous momentum due to the discovery of DNA sequencing technology by two different groups of scientists, by Sanger and the Maxim Gilbert. So, when people started to know that what is the composition of DNA, people thought that we now able to crack the mystery of the mother nature, that we know what we are made of. And then people thought that we will now able to know the mystery of the human genome, what is within us. So, a big ambitious project of human genome sequencing launched, and before the targeted time, we could able to achieve the sequencing of the entire human genome. And thanks to the more technological advancement, the engineering things, that we are able to know that the sequencing was now very much affordable. There are several different types of sequencing platforms are available. You will able to hear from couple of company persons like Illuminate sequencing, Quagen sequencing, Life Technologies, the Spiro sequencing. So, there are different first generation sequencing, second generation sequencing methods, and then recently the single molecule real time sequencing based on only one DNA sequence. You will hear this kind of talk also, the smart sequencing, then nanopore sequencing. So, these are different platforms to generate the sequence information data. So, this sequence information data when was so much available, so much cheap, and what happens, the cost of sequencing decreases, the more sequence was generated, and that actually flooded the sequence database. So, huge number of sequence information now deposited into the database. So, we thought that we will able to crack the mystery of the mother nature if we will do the sequencing. So, lots of huge number of sequence information generated. Then the question started coming, oh, we have lot of sequencing data, then what? who will how to the different organism different, because when we did sequencing information, we observed that all humans are similar, 99.99% we are similar, and in the human genome 95% of the genomic information is bogus, only 5% codes for important genes. So, if it is the situation, then how the organisms are different, and who will find out the function of all these proteins and how these sequences are related. So, this kind of questions started coming. So, how to analyze the sequence data? Sequence generation is not a problem, but how to really analyze the sequence data? Because what is this actually? Can anybody tell what is this? Yes, tell quickly. It is Egyptian hieroglyphs. So, are you able to read anything? So, for that you need some technique, some expertise to read this hieroglyph. Similarly, this is the sequence information, ATGC, only 4 letters. So, it is just like a boring hieroglyphs. So, if you do not have any tool to decode this information has no value. Okay. So, it was observed that again history repeats, that one subject, one individual, one institute is not possible to give solution to decode this information. So, again history repeats, and 4 major branch of science, biotechnology, information technology, computer science and statistics, they merge together to again give rise another subject called bioinformatics. Okay. So, this is the concept that how the subject bioinformatics evolved. So, when bioinformatics subject evolved, then the question, what is actually this bioinformatics? To know that, before we have to know what bioinformatics is not. If you understand what bioinformatics is not, then you will be able to realize what bioinformatics is. So, bioinformatics is not just using a computer to speed up biology. It is not just applying computer algorithm to biology, and it is not just an accountant of genomic data. Make it very clear that if you have a computer, if you have a software, you do a whatever thing is, it is not like that. So, what then bioinformatics? Bioinformatics is the creative use of the computer to define and solve the central biological puzzle. The computer becomes an hypothesis machine making prediction to be tested in the lab. So, only it is a hypothetical machine, it will assist you, it will guide you that how you can use the those information data to generate some knowledge. Okay. So, it is actually the marriage between the information science and the biological science. So, just like the central dogma of life, the bioinformatics subject also has a central dogma. This central dogma tells that if we have sequence similarity between 2 different sequences, if we have sequence similarity, then definitely there will be structural similarity. And if there is structural similarity, for example, this sequence similarity, if we have a query sequence, that means the sequence which you have generated, that is called the query sequence. So, if you have a query sequence and it is matching with some sequence, then if this matching sequence structure is known, that it is a zinc finger, for example, DNA binding protein, that zinc finger motif is there, then we can predict that query sequence as it is matching with that sequence. So, the query sequence should also have similar kind of structure. Okay. So, if there is a sequence similarity, there must be a structural similarity, and if there is a structural similarity, there must be a functional similarity. So, if this zinc finger motif is having the DNA binding activity and it is involved in transcription factor and other things, so the query sequence predicted to have the DNA binding activity, or it may also act as a transcription factor like that. Okay. So, if you have a sequence information, you can predict the function of that information. So, this is the power of the bioinformatics. Just by analyzing the sequence, we can able to know what is the function of that sequence. Clear? If it is not clear, you stop me, I can explain it more, because it is for the student centric program. So, definitely it is not just giving lecture from one side, you can stop me, ask me. So, so this is the central dogma of bioinformatics. So, bioinformatics subject, a huge subject, it is actually we can divide it into 3 basic components. The first component is development and implementation of tools and database. That is not our domain. That is the mainly domain of the computer scientists, mathematicians, and then development of new algorithm and statistical formulas, again computer scientists and the statistical persons. And third point is use of the tools to generate biological information and manage them. So, this is the domain which we are interested, that how we can use this sequence in. Yes.
Sorry for
Yeah.
to backslide.
Yeah.
Since the students haven't asked, I'll ask. I just wanted to ask the students, you should ask how much of the sequence should be similar to say to have the same function.
Yeah. Yeah, I'll be coming. So, just it is a central dogma. I will come now those complexities. Okay, definitely. So, bioinformatics. So, these are the 3 part, and our part is the last part, that is the using of those tools. So, now the title scheme. Till now it was only introduction part. So, now to have a test, have a to remove the fear that what I am going to talk, I have given a little bit introduction. So, this is the talk, that sequence based molecular phylogeny, and there I am not going to tell about the very complex things, just very basic concept and principles, so that you should understand what is those things. And because many times I have seen the students, even the faculties, even many people misuse, abuse different terms of bioinformatics, and after this training you should not do that thing. You should tell the correct terminology for correct thing. Okay? So, let us start. So, if you have done sequencing, many PhD students have done sequencing, they have done many advanced molecular work. So, you may come across, or you have generated different kind of sequence data. The sequence data may be small, maybe 1 gene sequence data, maybe relatively big, that is bigger genes, smaller genome of viruses, maybe big sequence data like the relatively bigger genomes of bacteria and other things, and whole genome, complex genome data of the eukaryotic genomes. Okay. So, these different kinds of sequence data you may have. To analyze those sequence information data, how we proceed. So, as it is student centric, so I will start from very basic, because there are many MSc students also involved. So, if you have a small sequence data, for example, the sequencing of small PCR product. So, if you do that sequencing from only one side, you generate a sequence information, you got a small sequence, that is okay. You may have, so just do a PCR and do sequencing, or you have many more few reads, a little bit bigger. So, you need sequencing from 2 ends of the PCR to generate the sequence, and in that case you will generate this kind of sequence. So, you have to find out a region which is overlapping between the 2 strands, and then you have to do. So, this sequence read is in this direction. So, you have to just make it in the reverse complementary direction of the sequence, and then join with the rest of the sequence to get a full sequence. So, in that way, instead of means sequence from 2 different sense, you will have the sequence of yes. Is this okay now? That's okay. Yeah. So, you will have only one sequence read. Now, you can do sequencing using a clone molecule. So, in this case, you can use a vector primer for the sequencing of your target genome, and when you will do the sequencing, what will happen? You will generate a sequence which will have the vector sequence also within it. So, many times I have observed that when you do sequencing, just raw data which you got, you submit it into the database. It should not be done. Before submission, you should carefully see that there should not be any vector contamination. Okay. So, there should not be any vector contamination into that. So, for that you have to remove the vector sequence from your sequence, and then this is the purified sequence which you can do analysis or any other things. Now, if it is a big sequence, large sequence data, then several such kind of fragments, sequence fragments will be generated, and several sequence data you have to do assembling of this overlapping sequence data, and ultimately join them to form this assembled sequence. Okay. So, for few such kind of things you can do manually by doing one by one sequence analysis, but if it is a whole genome, then three terminologies come. This terminology is called reads, contigs and scaffolds. Okay. So, a big sequence or a whole genome sequence generates several such kind of sequence data which called reads. So, they are the small sequence data of the big genome, they are called reads. When reads are aligned to form a complete sequence, they are called contigs, and these different contigs when you align you will get a scaffold. Okay. So, there are three terminologies, do not misuse or abuse these terminologies. Contigs, reads and the scaffolds are three different things. And for the whole genome sequencing, this contig assembly we cannot do it by manually, for that we need some sequence software, and these are different sequence software which is available. By these softwares you can do contig assembly and generates of the scaffolds. So, these are the actual sequence file, how it looks like. This is called chromatogram file or whole genome sequencing data. So, when this kind of data generated, then the question is what next? The next is to find out the meaning of the information hidden in the sequence. We need to compare this sequence with other sequence. Without comparing, we cannot able to know what sequence we have. So, if we want to compare them, then definitely the question is from where we will get the sequence to compare. For that, there are some sequence databases. So, from using those sequence databases, we can have the sequence of others and then we can compare those sequence. So, what is this biological sequence database? They are the organized bodies of persistent data. They have three basic components in any of this database. The components are update. You can update the database, it is not a closed database, it is a open database. So, every time, every seconds you can deposit anybody from the world, they can deposit their sequence to update the database. They must have a query component in the database, so that if you ask the database to give you a data, they can give you the data. So, there must be a query component and there must be a retrieve component, you can retrieve the data. So, this data biological databases are mainly of two types. One is called sequence database, second one is called structural database. Okay. So, structural database means here the structure of the proteins are deposited, this is called protein database. And the sequence database is again divided into two main part, one is sequence nucleotide sequence database and second one is protein sequence database. The nucleotide sequence database, there are three major players globally. One is called NCBI, EMBL and DDBJ. This is from USA, European countries and Japan, and these are again from the protein sequence database from European countries. So, they are different databases of not specific origin. But besides that, you may have several specific sequence database, for example, rice sequence database, any fungal sequence genome database like that, that you have specialized sequence database. These are general sequence database. So, these are the some short that this is called National Center for Biotechnology Information, NCBI database, European Bioinformatics Institute database, EMBL, and this is the DDBJ DNA data bank of Japan. So, these are the three global database. Now, the question may come that if I will take a sequence from one database or I will submit my sequence to one database, then whether I have to submit my sequence to all these database or whether I have to get one particular sequence from one database, another sequence from another database? No. These three major database, they have the understanding among themselves that any sequence database, if you deposit any sequence within 24 hours, that sequence will be available. You can retrieve the sequence from any database. Similarly, you can retrieve any sequence from any database that may not be deposited into that database. So, that is the understanding. So, with respect to the submission and taking out the data, there is no problem. Any database you can submit or retrieve. So, this is Swissprot. This is the PDB structural database where all the structures of the proteins are deposited. This is some specialized database like Ora sativa database. This is some fungal sequence database, Magnaporthi. So, like that several specialized sequence databases are also there. Now, the question is how to deposit those sequences into the database if you have a sequence. For that, there are several tools available in those databases. This GenBank is also called the NCBI database. So, they have two different options, one is called Bankit, one is called Sequin. If you have only one single sequence data, you have to submit through Bankit. If you have a large amount of sequence data, for example, cDNA data, EST data or something like that, you can submit it through Sequin. And for EMBL, you can submit it to Webin Align, that is the option in the EMBL database. So, this is the short that Bankit in the NCBI or Sequin. So, you can go there and submit your sequence. And for the genome sequence deposition, you can again use this kind of means option in the NCBI to deposit the whole genome sequence. But before submission of the sequence, you should do annotation of the sequence. So, again another terminology, what do you mean by annotation? Many times you may come across with such kind of terminologies. So, annotation, it refers to the commentary or explanation of the information appended to the DNA or protein sequence stored in the database. So, one information, one explanatory note or something which can define that sequence. Without that thing, again the sequence has no meaning. So, you have to tell the database this is that sequence from this organism. So, these are the different kinds of annotation that shows from where you are you have isolated that DNA, the country, the organism, the protein sequence, predicted protein structure and so many things you can use as the annotation of that sequence. So, that anybody when they can use those sequence, they can able to know that the information already the secondary information already attached to that particular sequence. So, this annotation is very much important. Before submission of the sequence, you must do annotation and then submit the sequence. So, you have sequenced gene or genome organism and deposit it into the sequence database. Now, the question what next? Next is to analyze those sequence. For that, you need the sequence. So, there are again terminologies, it is called data retrieval, okay? That is collection of data from database. Now, I will request every one of you in future, do not tell I have downloaded a sequence from database. It is I have retrieved a sequence from the database. So, what is the difference between download and retrieval? Can anybody tell? Anybody? What is the difference between download and retrieval?
Yeah. The basic English term itself should tell you.
That is the thing. What is the difference? Not clear? That is why you can frequently abuse this term, that download. So, for example, you have a file in the computer. You can, or if you have a file in your mail, you can download that file into your computer. Okay? But if I will tell that in that file download the page number 3, can you download the page number 3 only? You can open that file, you can copy that page number 3 and paste it into a new file. So, similarly, sequence database you cannot download. So, database is a kind of file and the sequence which are there, they are the pages of that database. Okay. So, one page you cannot download, one page you can copy, retrieve. Okay. Clear? The retrieval and download thing is clear. So, you cannot download the sequence, you can download the database. Okay. You can retrieve the information which is present in that database.
Sir,
yes.
But when we are using BLAST algorithm in the NCBI,
It is now written, earlier it was written retrieve. Now, they have also get confused, they wrote many mails, they wrote clarify, but people are not understanding. They are only asking and there are several mails also people got, this NCBI people got how to download the sequence. So, now they also got fed up and they changed, they also written download the sequence.
download sequence it
It is now, but earlier days when the sequence databases were there, it was only written as retrieve sequence, but people could not able to understand what is retrieve sequence. Actually, the basic thing is that you are actually retrieving, you are not downloading. Downloading can be done, the database it can be downloaded, because that is a file, but the pages, the database means a file and pages are the information, sequence information. Okay. So, you cannot means download one page of a file. Okay. Clear? So, now onwards you should not tell I have downloaded the sequence, sir I will download the sequence and do analysis, no. Okay. So, what we need to why we need to retrieve the sequence? To do two main things. One is genomics, and second one is proteomics. Genomics means understanding the interaction among genes, and proteomics means understanding the interaction among proteins. The genomics again can be divided into many things. The structural genomics means what is actually structural genomics. If you do the analysis, if you want to do a genome organization or what if prediction, what are the genes are there, then designing of the primer for detection and other things. So, this comes under the structural genomic component. Second point is comparative genomics, that is what if annotation, relationship of the variability and variability between the pathogens and their strains, molecular phylogeny. So, they comes under the comparative genomics. And then third component is functional genomics, that is prediction of motifs, blocks, domains in the protein, prediction of the functions of the protein, protein structural modeling and visualization of the 3D structure. So, these are coming under the functional genomics. And in the proteomics, the interaction of the proteins with other molecules and drug designing. So, these are the different components related to the plant pathogens and microorganisms, that why we need to retrieve the sequence to analyze these things. So, to do that thing we need to do some data mining, again another term. So, data retrieval means getting the information from the database, and second thing is data mining. Data mining means generation of the information from the data in the database. So, sequence data is there. So, now you want to generate the information from the data. For that you have to mine the data, and that is called why it is called data mining. Data mining can be done several ways. Here I am mainly today focusing only on this aspect, that molecular variability, phylogenetic, phylogeny and evolution. Besides that, we can find out the genes, primer designing, protein structure and function, promoter identification. So, these are other different types of data mining which we can do from the sequence data, but only I will focus only on this aspect, phylogeny and evolution. So, before going to tell about the phylogeny, just a little bit relax, because continuously if I will bombard, then you will also get bored. So, tell me what is this? Anybody can identify what is this? Bullet, correct. What is this? Pistol. So, this bullet is shoot from this pistol. Okay. So, what is this? This is the hole inside the pistol, the nozzle inside the pistol, it looks like that. They have carved things. So, bullet when comes out, it comes out as a rolling thing, and when it hits these structures, these are the raised structure. So, the bullet got injured by the structure and some scar marks developed onto the bullet. Similarly, bullet also gives some scar mark to the pistol nozzle. Okay. So, when first bullet shoot up from this, so these are the kind of scar mark it generates. So, when it shoot up from this hole, first bullet it got some scar mark into its body. Second bullet, the first scar mark was there, along with that some more scar mark also generated into the bullet. So, like that third and fourth if you generate, so they accumulate different scar marks. And the crime investigators, what they do, they try to analyze whether there is some commonality between these scar marks, and then they tell that yes, these all bullets shoot from the same pistol, because they are having some kind of similarity. How? Because these scar marks are similar kind of scar marks. Okay. So, during this shooting one by one, the variation is created here, and but there has some link. Similarly, if you see, this is the process of evolution, the shooting, and this is the variation in the individual, but they are originated from the same. So, they must have some phylogeny, they have some relationship. Based on that relationship, people can tell that they shoot from the same bullet. Similarly, as I told from the first slide that we originate from one single molecule. So, we must have through the process of evolution something which is there in the earlier days. So, that is the actually phylogenetics. So, what is phylogenetics? Phylogenetics is the science of establishing and analyzing the evolutionary relationship among the different organisms. How it is done? Analysis of the DNA sequence to help us understand the phylogeny of the organism. So, using the DNA sequence we can understand the phylogeny. And what is the purpose? The purpose is phylogenetic relationship help us to understand the evolutionary history of an organism and how they are related to each other. So, that is why we need the molecular phylogeny, to understand the variability, to understand the evolution, and to understand the relationship. So, what we know from molecular phylogeny? Whether our sequence has any homology with other sequence, how much similarity is there between our sequence and the sequence of others, and how our sequence phylogenetically related to other sequence. So, again 3 terminologies: homology, similarity, and phylogenetically relatedness. Okay. So, what does it mean? Whether they are same or different, because I have observed that students when they are giving thesis seminar and other things, they very infrequently misuse the term homology and similarity and phylogeny, and all those things is a khichdi. So, all this terminology when they have coined, they must have different meaning, not that it is everything is same. So, what is homology? Can anybody tell what is homology? Homology? Nobody? Huh? From the parent? Tell from the parent what? Any trait, yes. So, so this is a state of a gene or morphological character. You remember, it is a state of gene or morphological character that shares a common ancestry with different gene or morphological character. So, it is a state whether it is having ancestry with other sequence or not. For molecular sequence data, it is taken to mean that 2 sequences or even 2 characters within the sequence are descendant from a common ancestor or not. Okay. So, it is purely related to the ancestral relationship, whether that is there or not. Again, whether that is there or not. But what do we do? The term is frequently misused as a synonym for similar. We many times, every times we tell 2 sequences were 70% homologous. Okay. This is totally incorrect. Sequence shows a certain amount of similarity. From this similarity value, we can probably infer the sequence are homologous or not. Okay. So, similarity and homologous, two things are different. How different? Very good quote by Walter Fitch. What he told, homology is like pregnancy. You are either pregnant or not. A woman, can you tell a woman is 25% pregnant or 70% pregnant? Either it is she is pregnant or not pregnant. Similarly, the homology is the similar thing. It is a state, it is a qualitative character. We can tell whether 2 sequences are homologous or not. We cannot tell 25% homologous. If we tell 25% homologous, that is same as 25% pregnant. Okay. So, these are the terms we should clearly tell. If we are talking about homology, we will tell these 2 sequences are homologous or non-homologous sequence. You should not tell 70% homologous, never. We should not see, at least this batch of people, when they will give any seminar or any thesis seminar or anything, they should not tell 70% homologous. Okay. So, homology cannot be measured, only we can say whether homology is there or not, but we can measure the similarity. Homologous sequence must have similarity, but if there is similarity, we cannot say there is homology. Okay. If the sequences are homologous, then they must have similarity, but if two sequences are similar, 30% similar, they may not be homologous sequence. Okay? So, this is very clear, you should have this clear idea, homologous sequence must have similarity, but the opposite is not true. If there is similarity, we cannot say, because if you do any analysis, you may see that your sequence may match with some human sequence, some plant sequence, something, but you cannot tell that this plant sequence and your organism sequence are homologous. Okay? So, homologous things actually have two major families, homologous gene superfamilies. One is called orthologous gene superfamily and paralogous gene superfamily. So, yes.
How much similarity should be there when we say that two are
Coming. When I have asked this question, definitely I have to answer this question also, coming one by one. So, orthologous gene and paralogous gene. Okay. So, what do you mean by orthologous gene? Anybody knows? These are very basic things. Orthologous gene? No idea.
Which have originated from different parents, but still have a
Still have?
means that they are from different organism and have some
Hmm. So, for example, DNA polymerase of goat, DNA polymerase of human, they are originated from two different things, but their function is same kind of sequence, but they are found in different taxa. So, this is possible because of result of lineage transfer of the gene from one taxa to another during the process of evolution. And the paralogous gene, like hemoglobin and myoglobin, it is from this one taxa, two different gene, two different function, but they have the similar kind of sequence. So, like hemoglobin and myoglobin, they are having same thing, but one is transferring the oxygen to the blood, another one is in muscle. Okay, two different function, but originated from the same thing. So, this kind of things is a result of gene duplication. So, during the process of evolution, two things happens. One is lineage transfer of the genes or sequence, and duplication of the genes and sequence in the database, I mean in the organism. So, due to that, the variation and other things created, and you have the commonality as well as the difference between the different organisms.
Sir, what is the meaning of lineage transfer?
Lineage transfer means from one means taxa during the process of evolution, it transfer to, this is a parallel, means evolutionary pathway, it is going to another taxa during the process of evolution. So, it is, you cannot say that within 1 days or 1 generation it transferred. Okay. It is a huge, it is a millions of years is transfer from one taxa to another taxa. So,
when we say that the genes that got transferred through horizontal gene transfers, are they orthologous genes?
horizontal genes, yes, those if it is transferred from one organism to another organism, that is, those are the genes which is transferred across the organism, and that those are the orthologous genes actually.
Sir, but initially if two genes were transferred horizontally, but due to some reasons in the different organisms, now their functions have changed, so then they cannot be considered as orthologous.
Now, function, how it will change? If the sequence are same, they will do the similar kind of function. That is the things. If the sequence are same, they will do the similar kind of function. Definitely, sequence same, it is not 100% similar sequence. It cannot be possible, because during the process of evolution, those mutation, those changes will definitely takes place. But if you see the broader sense, the DNA polymerase of goat and human, they must have the similar sequence. Then only the polymerase function is there. Otherwise, it will not be there. So, some motifs will be there which is conserved across the families, across the genera. Okay. So, if we have a sequence, then our target is to find the homology. This is our main target. To find out homology, what madam has told, how to know that how much sequence similarity should be there to find out homology. So, we need to find out similarity or identity to probably infer whether homology is there or not. So, similarity or identity we have to find out, and for that again these two terminologies, similarity and identity. Sir, I do not know how we will finish by 15 minutes. So, similarity or identity, again two terminologies. For example, this is one sequence and this is another sequence. Okay. We would like to find out what is identity and what is similarity. So, if you see these are the 3 sequence in this 2 sequence, these are the 3 nucleotides which is exactly matching. Okay. So, this is called identical match or identity. So, if it is exactly matching, these 2 are similar match, purine to purine, pyrimidine to pyrimidine, and these 2 are mismatch. Okay. So, there are 3 kind of situation is possible if you try to analyze 2 sequence: identical sequence, similar sequence, and mismatch sequence. Okay. And how we are actually doing these things? We are doing a positional similarity or identity search, that first nucleotide is what in this first, second is what in second. So, this positional similarity or identity we are searching. Okay. And this finding out the positional similarity or identity is called alignment. Okay. Because very commonly we use this term alignment. What is alignment? Alignment is the identification of positional similarity or identity. So, when you find out this alignment, then we can tell that 2 sequence is how much similar, how much identical, how much non-identical or mismatch. And if mismatch is more, then they are not homologous definitely. If identity is more identity means there is more probability of 2 sequence being homologous. So, this alignment is the computational hypothesis which identify the positional similarity or identity between bases and amino acids, and this alignment of 2 type, one is called global alignment and second one is called local alignment. So, what is global alignment and local alignment? For example, if this is one sequence and another sequence is there, but in these 2 sequence there is only one stretch of sequence which is showing similarity or identity with the other sequence. The other stretch of sequence of both the things are not showing similarity or identity. So, only a stretch of sequence is showing similarity or identity. So, in that case this is called local alignment. So, local alignment try to find out the stretches of sequence which is showing more similarity or identity. The stretches of sequence where mismatch is more, it will not show. It will only show the region where it shares much more similarity or identity. But for global alignment, if you do, then it starts from first nucleotide of the first sequence and first nucleotide of the second sequence, and they do the entire alignment. There may be some gaps, there may not be all the sequence are showing similarity, but it will do a alignment based on the entire sequence stretch. So, local alignment is a very quick thing to know that in the entire sequence how much region is showing similarity. But if you really try to tell what is the percent similarity or percent identity, then you have to take entire sequence, the entire coverage. If you not take the entire coverage, you cannot tell the exact number of identity or similarity, how I am coming later. So, how to align these sequences? For example, we are trying to align these 2 sequence. There are 2 match sequence, 3 mismatch sequence, and there are 2 gaps. Okay? This kind of sequence, a very small sequence I am showing you. So, these 2 gaps means this sequence is 2 nucleotides shorter. Okay? So, how our main intention for this alignment is how we can increase the match sequence and reduce the mismatch sequence. And if there is some gaps, how cleverly we can put these gaps in such a way so that they can have this much more match sequence. So, we can do like this, these 2 gaps we can put here, and here we have increased the match as 3, mismatch 2, again the gap is 2 only. Or we can do like this, these 2 gaps we can create it here, and then you can see there are 5 match, there is no mismatch, and there is 2 gaps. Okay? So, in that way we can increase the mismatch. So, among these 3 different alignment, this alignment is much more perfect because it is reduced the mismatch. Okay? And this is the alignment. But you remember these gaps we have done, actually you do not know whether these gaps are actually there or not. So, you are tampering with the data, you are manipulating your data. So, if you are manipulating, you have to give some penalty. Okay? So, if you put one gap, you have to give one penalty. If you put another gap in another the region, you have to give another penalty. But if you put a gap first and then you put another gap just after that, then the penalty will not that much, because already one manipulation you have done in that region, you can do another more manipulation. Okay? So, due to these things, so what actually have done? So, if this match is 2, if you give a means number or value of this alignment 2 and this alignment value as 3, this has 5, but as you have put 2 gaps here, then 2 marks will be reduced from here or 1 marks will be reduced from here. So, you have done, you have got the correct answer, but you have cheated. So, 1 marks will be deducted. Okay? So, like that some score value will be generated, and the more score value means more probability of the correct alignment. And as you are doing this kind of gap, so you have to give some penalty. So, this is called open gap penalty. When you open a gap, that is called open gap, and when you extend that gap with another gap, that is called extended gap penalty. So, if you do, anybody has done sequence alignment, you will see that it is written that how much open gap penalty was there, how much extended gap penalty was there. So, that means to do the proper alignment, you have to give some penalty, and then the correct alignment will come and the score value will come. But it is only 5 nucleotides, so many alignment is possible. If it is a huge large sequence and large number of sequence, only 2 sequence we are doing with 5 nucleotide, and so many possibilities there. And if it is 1000 nucleotide, or if it is a genome, and it is like that, if you have thousands of genome, then how to do those analysis by manually, it is possible? No. For that we need dynamic algorithm or programming. Those programming will do alignment for you and tell you the result. What result it will give? The programming generates some scores, these points, that if you some penalty and other things, alignment has how much point, that is called score. So, more score indicates more similarity, and they also generates a E value. This E value means the probability of not matching the sequence with the other sequence. So, if E value is 0.003 means the probability of not matching your sequence with the sequence which is deposited is 0.003. That means the probability of matching is 99.99 like that. Okay? So, more score means more similarity, less E value means probability of being homologous sequence is more. Okay, clear? So, there are 2 terms again, scores and E values. So, if the E value is less, that means there is more probability of those 2 sequence being homologous. Okay. So, this is the kind of score the softwares actually generates, the different value points. So, minus means there are more gaps they have created, so score is minus. Okay. So, this is a kind of blossom matrix used by the different software. So, the sequence alignment tools to do these things, what are the different softwares or programs which we use? These are the different programs: BLAST, FASTA, Bleach, Beauty, ClustalW, Mega. So, these are the different softwares or online programming which we do to do the basic alignment. Now, what is BLAST? It is not bomb blast. Yes. How do we create or software create?
Why is there a need of creating a gap? This is for them to see that the 2 sequences are similar or dissimilar.
Yes.
So, if it is aligning in a normal fashion, so it is going to see that the 2 sequences are same or different.
Yes.
Then why are you trying to align?
Yes. Now, yes. We have these 2 sequence. Now, this sequence when I have done, we got in this sequence we got only 5 nucleotide. Here we got 7 nucleotide. Okay. That means the first nucleotide is 2 nucleotide shorter. Now, these 2 nucleotide, that means there is a mutation or deletion from that. Now, deletion has taken place at the last, or deletion may taken place here, or deletion may taken place here, anywhere it ultimately when sequence comes, it is continuous sequence, but during the process of evolution that 2 nucleotide deletion may take place from here also. So, it has actually it is coming like that, if the 2 nucleotide deleted from here, then only they are matching properly. That means, it may be possible during the process of evolution the gaps has created in this region, not at the end, because sequence when you read it is continuous, but actually during the process of evolution where that gaps has created, that we also do not know. That is the probability that that gap may be created here, may be created here during the process of evolution. And the more, how we will know that yes gap has created where, because if you have more match sequence, then the probability is more that the actual during the process of evolution the gap was created here, because then only the more match is coming. Clear?
What if it has not taken like that and
Then this is the condition. If you directly think that only the gap has been created at the end, then the match is less, mismatch is more, then your inference is that these 2 sequences are non-homologous. Okay. So, you have to, because not that all the sequence will be in the same length, okay. There will be different size of sequence. So, if you are not able to, that means in different sequence different deletion has taken place, or in another word in other sequence different addition has taken place. So, where that addition has taken place, where the deletion has taken place, if you do not take that into consideration, your analysis will not be proper. Clear? So, sir, what I will do? I will continue because it takes. So,
these are the points which are very required for the students.
that is why for the students.
If you want, we can give a break for tea now.
You can give a.
We can take another half an hour, maybe up to 12 o'clock.
Yes.
No problem.
Because this basic information, they are doing sequence analysis, they are doing all those things without knowing the basic. Blast, sir, I have blasted the sequence. Hari blast, a bomb blast, I have blasted the sequence. Blast is the name of an analysis. I have blasted the sequence. I have heard that students are telling like that. I have blasted the sequence and got this. Blasted means they think that it is a bomb blast. So, tea break will have?
Yeah, we'll have a tea break.
Okay.
For
Yeah.
all the trainees, please, tea is served outside. Have tea and come back.
So, are you thinking 5 minutes? Are you thinking it is interesting? Many pathology students I have give lecture to this same lecture to class and other things they may know, but other students for them that may be. These are not very proper. I have not done. No. That is format for the analyzing the sequence in the FASTA software. FASTA is a format. For example, there are different formats to analyze the sequence in different softwares. So, for analysis the sequence in FASTA software, you need the FASTA format sequence. But now FASTA is so common that FASTA can be taken up by any of those things.
There is a software called Blast to Go. It shows like sequence blasting
Because for the students, the people are now started to thinking about the student term only, the researcher term, because we are not the statistician or we are not the computer scientist. So, many such kind of things happens like that, people now talk in the very layman language that blasted the sequence. But blasted means that is not bomb blasting, that blasted the sequence. Okay. But you should know the proper terminology, then it will be very much useful. So, these are the sequence alignment tools: BLAST, FASTA, BLEACH, BEAUTY, CLUSTAL W mega, which is used to do alignment. Now, I got many inquiry during the tea time that how to do really those things, how to do this alignment, how to purify the sequence, to remove the vector and other things. So, we will have a practical on this on 24th. Sir, on 24th? So, we will have a practical definitely where you will bring the laptops and we will show those things how to do. So, starting with the BLAST, it is not bomb blast, it is the acronym of a big thing which is called Basic Local Search Tool, that is the BLAST. So, it terms tells us that it is doing a local alignment, it is not taking the entire sequence, it will just randomly see that the region which is showing a stretch of the region which is showing highest similarity or identity. Based on that thing, it will tell you that how much similarity is there. So, what it does, BLAST is the algorithm used by a family of 5 programs that will align your query sequence against the sequence in a molecular database. So, whatever sequence you will give, it will do alignment, online tool actually it is. So, if you just give your sequence and you, there is a button called BLAST, you do the BLAST and then it will do with the 5 different programs and it will tell you with which stretch of sequence of your sequence is matching with what sequence. Statistical methods are applied to judge the significance of this method, that which one will be the most significant and which one will be the least, and alignments are reported in order of the significance as estimated by the applied statistics. So, the most means confident sequence or significant sequence will be giving you the first, then one by one it will come. So, BLAST actually has different programming. This is the BLAST N, which compares a nucleotide query sequence against a nucleotide query sequence database. So, if you want to give that software the DNA sequence, it will align the DNA sequence of different organism, different genes deposited in the database. So, it is called BLAST N. BLAST P, it compares amino acid query sequence against amino acid query sequence. So, if you want to analyze the protein sequence, the amino acid sequence, then you have to give the option BLAST P. So, in the BLAST, these options will be there, BLAST N, BLAST P, like that. Then third one is BLAST X. This compares the 6-frame conceptual translation product of a nucleotide query sequence against a protein sequence database. So, you give the nucleotide sequence, what it will do? It will conceptually translate that protein sequence into 6 frames, 3 frames from one strand, 3 frames from another strand. So, total 6 frames it will generate the amino acid sequence. Then all these 6 frames amino acid sequence, they will searched along with the protein sequence database. So, it is the nucleotide protein sequence matching. Next one is T-BLAST-N, it compares a protein query sequence database to the nucleotide database. So, if you have a amino acid sequence, you want to know that particular protein is coming from which gene. So, for that you have to do T-BLAST-N. So, now in this case the amino acid sequence, you want to know what is the corresponding gene for that sequence. So, it will do the opposite thing, it will compare the protein query sequence against the nucleotide sequence database, and then it will show you the significant matching. So, this is the how it looks like. So, you have to give your query sequence here in the things, and there will be one option called BLAST. So, it will BLAST and this will generate the results in this. So, you can see they will generate the result as maximum score, total score, query coverage and E value. This query coverage means in the entire sequence how much region it has covered for the analysis. Okay? And this is the score. Based on the score, actually they have organized that which one will be the highest score. That means this has the most searched means similarity is much more with this particular sequence. And the E value, if it is 0 means the exact sequence you have, and if it is means less, that means it is more probable that this sequence is with homology with that sequence. So, these are the this is the result which the BLAST will actually show you. Then what we actually know from the BLAST? We know that sequences that share similarity with the query sequence, and it helps us to retrieve those sequence. So, BLAST is only for these two purpose mainly, that just at a glance to know what sequences my sequences is showing similarity, and then you just retrieve those sequence from that BLAST analysis. Clear? But you cannot do a quantify the sequence similarity as it is the local alignment. You cannot quantify the sequence similarity, though BLAST also generates one column which is showing similarity, this much percentage, that much percentage, but that percentage is only the region which it searched. The rest of the region which is not taking count, it will not tell you, and cannot tell us about the relationship between those sequence. As it is only local alignment, it cannot tell you the entire relationship of the entire sequence. To explain that thing, how I am trying to tell that, suppose this is the sequence result with your query, this is the subject which is deposited in the database. So, this is your query sequence, and in the deposited sequence this has matching, or this is the deposited sequence, your query sequence is matching in this at the different stretches. Okay. So, in the entire query, in the entire coverage is very high, because it is almost taking the entire sequence, and if in this case you see here it is showing 92% similarity or identity, because it has taken out of 373, 346 is the identity. So, that is okay, because it is almost taking the entire coverage, but it has not totally coverage, it is not totally 373, some nucleotide it has not taken, but if you take that also, that will not change very much this value. Okay. So, in this case if it is 92%, it is we can very comfortably tell that okay, 92% means query coverage is also very high, and the similarity also high. So, this two sequence probably the homologous sequence, but this sequence, this kind of things may situation 2, less query coverage, but high sequence identity. For example, these are the two sequence we have taken. So, this is the deposited sequence, and this is in the entire sequence only this stretch is matching with this, and here you can tell this is also showing 91% similarity, but the entire sequence which you have given, suppose it is 373 nucleotide, it is only showing 84 stretches of sequence out of the 373 nucleotide, only 84 nucleotide stretches is showing the similarity, rest of the things are not showing, but BLAST will tell you again 91% is the sequence similarity, but it is only within that stretch which is analyzed, not the other things which it has not analyzed. So, just by taking this value, the similarity or identity value, both the things, that means same. The first sequence which it is showing the coverage 100% and similarity is 92%, and here coverage is only 10% or 5%, but identity is 95%, you cannot tell that both the things results are same. Can you tell? So, you cannot tell by this way. So, in the earlier case, those two sequence may be homologous sequence, but in this case they are not homologous sequence, though it is showing in this stretch it is showing this much percent similarity, but they are not homologous sequence. Clear? So, you have to take, when you do a BLAST analysis, you have to take those sequences where the query coverage is very high and sequence identity is also very high. You take those sequences and then do analysis. Clear?
Sir,
Yes.
E value, sir. E value.
E value means that not this, the probability of not matching is the E value. So, if the E value is less, means probability of matching is more. Okay. Probability of matching is more means there are much more try to be the homologous sequence, and if the E value is high, that means probability of matching is less. Okay. It is inversely proportional.
And in some cases it is minus for ideal condition.
If it is exactly matched, then it is 0. Okay. Otherwise, it will have means always it is written 10 to the power minus 3. It is the log value actually. That is why it is inversely proportional. Okay. This E value is the log value actually. That is why it is inversely proportional. It is clear that that means from the BLAST never, I have seen many students just take the identity value from the BLAST and tell that this is the sequence identity. You have done? Many students has done like that. Just take that sequence, the whatever BLAST result is there, the last column is a percent identity, take that identity value and tell that this is the sequence identity. No, that is not the sequence identity, because that is the local alignment sequence identity. So, to understand the real sequence identity, we need to do global alignment, not the local alignment. Local alignment only to have a initial idea that with which sequence our sequence is showing similarity and retrieve them. And then after retrieving, you have to do a global alignment to exactly know what is the percent of identity or percent of similarity. So, so global alignment is done in 3 different steps. The first step, it does a quick pairwise alignment. What the BLAST has done, that is the local alignment, but it will do a global pairwise alignment, 2 sequenced at a time. They do pairwise alignment, then they form a guide tree, a relationship tree they will form, and then they do a progressive alignment following this guide tree. So, this is the kind of condition if you do. So, you can say this sequence with this sequence, they have done a pairwise alignment, they generated a score. Again this sequence with another sequence, they have done a alignment, generated a score, and after that they do a progressive alignment. So, they found that, for example, if you have given 30 sequences to do analysis, they found number 21 sequence and number 6 sequence, they are having highest score. So, they took these 2 as one group like this. So, they created one group with these 2 sequence, and then they analyzed with this group with other sequences, that with this group what group is showing much more close similarity, and again they generate another scores. So, based on this scores, now they reveal this sequence as value, percentage identities, as well as you can generate a relationship. So, it will, so ultimately it will tell you this kind of alignment, and then it will generate a kind of percent sequence identities, and it will generate a relationship tree. So, this is called the molecular phylogenetic tree. So, what is this molecular phylogenetic tree tells actually? So, it looks like this. Okay? So, what is these things actually? So, there are 3 components. One is this kind of area from where different branches are coming out, a joining, a point. So, this is called nodes. So, these nodes are the taxonomic unit. Okay? Nodes are the taxonomic unit from where different branches generate. This is called branch. This actually tells us the relationship, and this is the length. This tells us the genetic distance. Okay? So, there are 3 things. One is length of the branch, one is the branch itself, and this is the nodes, okay, or clad, we call them clad also. And there are some value which is given just below or above these nodes. This is called bootstrap value. This tells you the significance of this grouping, whether they are much more significant or not. The branching pattern of a tree is called the topology. The how this tree is branched, that is called the topology, and the relationship through this line is called dendrogram. So, we call this phylogenetic tree as dendrogram because we are presenting the relationship through lines, and that is why it is called dendrogram. Now, this tree are of 2 types. One is called phylogram, one is called cladogram. Why I am telling those things? Because when you do a sequence alignment or analysis, often not always, you do not care about what is this phylogram, cladogram, or what kind of things. You just click software, it will give you a tree, and you are very happy that you got a tree. But what for? Because in the software everything is written, phylogram, cladogram, slanted cladogram, several, some branching. So, you think that yeah, I have to give a very good picture. So, sometime in goal picture, sometime you give all those kind of beautiful pictures without having understanding that what is the requirement of you. Because software, if you click one, software will give you a result. That is why first slide I told, it is not just the use of the computer, it is the use of creative use of the computer to solve the problem. So, if your intention is to group a pathogen, to do a taxonomic grouping, you do one type of tree. If you do the grouping based on your evolutionary data, based on your evolutionary pattern, then do another grouping. Not that it is for beautification. Okay. So, there are two major type of tree, one is called phylogram, one is called cladogram. Okay. And both this tree again could be represented by two ways. One is called rooted tree, another one is called unrooted tree. So, many times I have seen some goal picture, some terra picture, some square type of picture, some different things, but what for those things? What is those meaning? Phylogram, cladogram, rooted tree, unrooted tree, what does it mean? So, from now onwards, after this training, the trainees should not misuse or abuse this terminology. For what purpose you are using, you should be very much clear that my purpose is this. So, I have to use only this type of tree, not other type of tree. So, phylogram means this is a phylogenetic tree that indicates the relationship between the taxa and also conveys a sense of time or rate of evolution. So, phylogram means not only the relationship, but also it conveys a sense of time or evolutionary thing, that how they are evolutionary related. The temporal aspect of the phylogram is missing from cladogram. So, cladogram is the tree where this temporal concept is not there, that means the time concept is not there, but in the phylogram the time concept is there. So, how actually the phylogram looks like? The phylogram looks like this. Many times if somebody, some PhD student has done the sequence analysis, you may have seen that some trees are looking like that, some branches are big, some branches are small. So, tree is not very even. Okay. So, some are very going, some are not going very up. So, what does it mean, this kind of tree? Some are very branching is going ahead, some are not, some are very much branched.
Also that line is not complete.
Line is not complete. So, like that this kind of things you will see. So, that means, so if you see this kind of grouping which is going further than a common line, if you draw a common line here, it is going ahead. So, these are the evolutionary more advanced branch. So, the organism, the sequence which you are analyzing, if they group together, they are grouped together means they are phylogenetically related, as well as they are evolutionary much more advanced, because it is going, because here a time concept is there. So, if your sequence you are not doing for any time, you do not have any time scale, if you do that thing, it will generate some result, but you cannot interpret. You cannot interpret what for this things is going ahead, but if you can give a time, and if you can put that this organism, this group of organisms evolved during the Neolithic stage and during this stage, that stage, then you can probably tell that due to that evolution it is going advanced. Now, this is another branch. Here the evolutionary they are more conserved, because they are going as one after million of years. So, this is now time. So, after million of years they branched it into two, and after that also it is going, it is not having much more diversity in this group. So, they are evolutionary much more conserved, and if you have this kind of pattern, very quickly they started branching though. So, they are evolutionary much more diverse, more changes taking place. Okay. So, if you want to do your analysis with this point, then you do phylogram. If you do not have this kind of data with you, only the sequence data you have, you just tell the software to develop phylogram, it will develop a phylogram, but you cannot tell or you cannot infer anything from that data, because you do not have any time concept in that. But if you have time, also put it in, means your data is also related to the time. For example, you are analyzing some microorganism from some fossil, something like that, and you know that carbon dating this fossil is from this age, then you can do analysis, and then you can tell that how that evolution takes place in that particular organism and other things, then you can, that is justified to use the phylogram. But if you do not have that thing, do not do phylogram for the sake of beautification. So, cladogram, the same thing here, time concept is not there. As the time concept is not there, so all these things are in one line. So, all the sequence, if you see, they will be just staggered at one point, and all the branch is there, because there is no concept of time here. So, this is called rectangular cladogram or phenogram. there is another which is called slanted cladogram. Okay. There are two types of cladogram, rectangular cladogram and slanted cladogram. Again, what for these two? Many students use this kind of things without knowing what for this is required. So, rectangular cladogram is required when we want to do grouping for the taxonomic studies. Just to group for the taxonomic study, then only use this rectangular cladogram, do not use slanted cladogram. Slanted cladogram again is used to do the evolutionary studies. It is not related to the time, but in the evolutionary study there are many aspects. For example, ape and human started their journey from one point, then they separated out. So, they diverged out. Okay. Human and rice also originate, but they originate independently and they are growing. So, this kind of evolutionary thing is called how the origin evolves. One is divergent origin, one is parallel origin. The two organisms develop parallelly. So, they are called parallelism. Two organisms once upon a time they were same, then diverged out. So, divergence. It may be possible two organisms due to the mutation and mutation they may converge also in future of time. So, to understand this kind of things, convergence, divergence and parallelism, we need to use this slanted cladogram, not this. But if it is only taxonomic grouping, most of us mainly do this, only try to restrict rectangular cladogram, not the slanted cladogram. Slanted cladogram means when you want to know convergence, divergence or parallelism in the evolutionary study, then you use that thing, because this is kind of convergence that in future these two sequences, though they are originally from two, but due to the continuous mutation in future they may converge and again gives one single entity. This is called parallelism. Parallelly they are evolving. This is the divergence, that it is started and then diverge, again going diverge. So, it is divergence. So, if you want to explain your data with this kind of things, then use this. If you don't want to explain your data with this point, what for you are using then? Okay. So, based on your interpretation, your data, you have to choose what tree you will put it in your thesis or result or anything. Okay. So, do not take for beautification purpose, only for the purpose that whether you can explain your result, support your result with this type of tree, then only use that thing. Otherwise, whatever purpose you use for that purpose.
The two parallel lines that you are talking about, the same parallel line when you have three components, that is also divergence.
So, that is diverse from here. So, in this process of evolution they are diverging, but they are independently, they are parallelly evolving. They are parallelly evolving from one thing.
So, the situation is not mutually exclusive also. Please remember that when you are having this kind of a data, the same data when you start at the same time, parallelly two components evolve, but at some point of time they also have three different divergences also.
Three different divergences, but they are also evolving parallelly.
Sir, in parallelism, what is the similarity between these two parallel evolutions?
That is this parallel evolution actually the means, that is why the lineage transfer of the gene takes place, because it is everything is there in one, then it comes out from, due to some mutation it comes out from one. So, the genes which they have, they also carrying that thing, again after few million years they also evolve like that. So, in that way what happens, they carry the same linear genes and they are present in different axes. Okay. So, yes.
In this case where there are three, firstly they are diverging and then they are moving parallelly.
Yes.
So, if the three parallel lines are closer or they are more far, so what is this going to indicate?
This length is not having that means this length, that has no, because that you can in the software you can increase that thing, only the length, not the height. Height you can change, height has no meaning on this, only the length has the meaning, not the height. So, either this height or this height has not that meaning, it is the meaning is these lines, the parallel means horizontal line, not the vertical line. Okay. So, the tree can be presented as rooted dendrogram or unrooted dendrogram. Rooted dendrogram means the evolution started from one particular parent and then they started evolving, and this is unrooted dendrogram where there is no root. If you want to study the evolutionary relationship between human, rice, mosquito, tiger, so you cannot think that they may have one ancestor. Okay. But if you want to understand the evolution of different bigomoviruses, then all the bigomoviruses must have coming from one bigomovirus. So, in that case you have a rooted dendrogram. So, again depending upon your data set, if your data set do not have a probable means ancestor parent, you use this, but if it is having, you do not have to use this. Many students goal map. This goal map means that is unrooted. You are trying to do a Magnaporthi sequence analysis, goal. Why? All the Magnaporthi different isolate may coming from one particular ancestor. Okay, then why goal? That means they do not have any common ancestor. But definitely if you want to do Magnaporthi and other different fungus, then also this kind of things is okay. But if one organism, one particular thing, different isolates you are using, definitely you have to use rooted tree.
One genus, many isolates are strange.
Okay. So, why I am trying to tell, because software you click that kind of thing rooted, it will give root. If it is unrooted, software will not dictate you. You have to dictate the software what you want. So, assume that all taxa derived from a common ancestor and assume that all taxa do not have a common ancestor. Then there are two different methods of generating this phylogenetic tree. One is called character based method, another one is called distance based method. Character based method means, suppose these two are the sequence, we are taking individual nucleotide wise means analysis. This T is mutated to G, so it will generate a score value and then it will generate a matrix and then it will generate a tree. But the distance based matrix means, suppose these two sequences are there, one is T, one is G, another sequence you have added, here also T. That does not mean that this sequence is from here. It may be possible that T is there because the original thing is there, then it is score 0, but it may be possible that one generation it was G, then next generation again it revert back the mutation to go back to this. So, due to this generation process, there is a score is 0, but the genetical distance is 1. Okay? So, that may be possible. So, different software can analyze that kind of distance based approach and then they will generate again a distance matrix and then they generate tree. So, there are character based approach and distance based approach. So, these are important character based softwares or algorithms which we use: maximum parsimony, maximum likelihood. When we use maximum parsimony, again software will not tell you what you have to choose. In the software you will have maximum parsimony approach, maximum likelihood approach, you have to tell the software which you want. Now, if your sequences are quite similar, different strains or isolate of a particular pathogen, and if you have relatively less number of sequence, then you use maximum parsimony method. If your sequence is much more diverse and much more intense, then you do the maximum likelihood method. Okay? So, if you are trying to analyze, for example, in our case I want to analyze a potivirus, a bigomovirus, another virus. So, I will use maximum likelihood and parsimony when I will use the different isolates of bigomovirus. Okay? So, when it is very much close, because close means almost all the nucleotides will be same, only few nucleotides will be different. Clear? So, character based approach is perfect for that. Maximum likelihood means maximum nucleotides are different, only few nucleotides will be matching. So, again the character based approach is there. Okay? But if it is in between, that some of the sequence stretch is matching, some of the stretch is not matching, then we have to use distance based method, and that is I am coming. So, this character based method just one example how the software does actually. For example, I want to analyze human, rabbit and chicken, this thing. So, 1 mutation, 2 mutation, 3 like that. So, it generates a score between human and rabbit 1, between human and chicken 3, and between rabbit and chicken again 3. 3 mutation, 3 score. Very easily I am telling, it is not like that, software tells so many things, but for your understanding very easily I am telling. So, if that way score will create. So, if the score is creating, then what, how the tree is developing, that means this human and rabbit, our score is 1. That means the least mutation is taking place. So, that means they are much more closely linked. If much more mutation is there in this case, that means they are much more distant. If less mutation is there, they are much more close, maximum parsimony, they are maximally parsimonious, close together. So, human and rabbit are close. So, you take human and rabbit 0.5, 0.5. So, this is the 1. So, you divide that thing and then you join with the chicken. So, this is 1.5 and 1 and this 0.5, that means 1.5 and 1.5 3, human and chicken is 3, then rabbit again 0.5, 1 and 1.5, again rabbit and chicken 3. Okay. So, in that way they generate the character based approach generates the tree. For the distance based approach, the example is neighborhood joining approach. So, how, for example, I want to study spinach, rice, mosquito, monkey, human, different things. Okay. So, a distance matrix will be created by the software and we will now find out which one is mostly related. For example, here the distance is very less, 3.3, that means these two, monkey and human, they are closely related. So, first step we make a group monkey human, make a group monkey human. The second step we have to understand with this monkey human group which one is closely related. So, again the software will generate another table next cycle. So, now it will take monkey human as a group and try to analyze with this sequence, and then with this group they try to find out and they observe that with monkey human mosquito is coming closer. So, now they join with the mosquito. So, the branch is growing and then mosquito monkey human they will find is as a group and then they will try to analyze with what they are more. Now, another cycle, but here we observe that it is not between monkey and human, the close relationship is between rice and spinach. That means there is another evolutionary group. So, this is called polythetic origin of group. So, this is one origin, this is another origin. So, spinach rice will be one origin, this will be another origin. Now, they will put together to form another the super group. So, this is one clade which is having this is second clade and these are parallel evolution and this is divergent evolution like that. So, you can now explain. So, how the software actually does this analysis and generates the tree. Clear? You do not have to do, you just have to click, but you have to choose whether you need a distance based method, whether you need a character based method, that you have to choose, but software will generate the result for you and you have to now infer that what actually you want to say. The last thing is that you have to do bootstrapping. Bootstrapping means judging the confidence. Suppose if you have a group of people and I told you, you make your partner pair, you make your pair what is ever available, and for example, some boy is pair with one girl, now another girl will come, he will leave that girl, join with another girl. Okay? So, bootstrap will actually tell how much is really they are having the chemistry to stay together, and if they do not have the high bootstrapping, that means if any another girl will come, that boy will go. Okay. So, bootstrap will tell that okay, today I have taken 10 sequence to do a phylogenetic analysis to make a grouping. If tomorrow 11th sequence will come, whether that 11th sequence will disturb this harmony or not. Okay. So, this bootstrap value is a method to assessing the confidence of that grouping. Okay. If the grouping confidence is very low, that means these two sequences today may be staying together, but if any new sequence will come, they may separate out and they may form another group. Okay. So, they will judge these things by doing the continuous bootstrapping, means 10 times or 100 times or 1000 times they will analyze the sequence, taking out one sequence. Suppose you have given 10 sequence, they will analyze one sequence with 9, then they remove one sequence, then with 9 sequence they again do the analysis, and in that way they do bootstrapping. जैसे bootstrap करते हैं, so कितना बार कैसे करोगे और हर बार एक ही टाइप का हो रहा है क्या नहीं, and then they will tell that whether the two pair which are coming together, how much after 100 times or 1000 times if you do, how many times is the probability that they will come together. So, the value will be 90, or 90 means in 100 times 90 times they are coming together. If it is 10, that means in 100 times only 10 times they may come together. So, there may be possibility if any other partner will come, that group will break. Okay. So, final, there should not be any confusion. So, if you have a set of sequence data, then what you have to do? You have to obtain a multiple alignment using any software, and after obtaining multiple alignment you have to ask the question that whether there is a strong sequence similarity or not. If there is a strong sequence similarity, then if it is yes, then you do maximum parsimony method, because strong sequence similarity means they are close very. If they are close, only 1 2 nucleotide difference, you have to do the maximum parsimony method, and then do bootstrapping and generate the tree. If it is not, if the sequence similarity is not strong, then the question is there is clearly sequence similarity? The sequence similarity is clear or it is totally different? If the question is yes, it is clear, 60% or 50% or 70% similarity, not the 95% similarity, but 60-70% similarity is there, then go for distance based method, that is the neighborhood joining method. Then again do the bootstrapping and generate the tree. And if there is not at all strong similarity, only 20%, 30% similarity, so much more sequence is diverse, only few sequences similar, again it is character based approach, do a maximum likelihood method and generates the bootstrap and generates the tree. Clear? So, depending upon your data set, you have to choose whether you will do maximum parsimony method, maximum likelihood method, or this neighborhood joining method. Not that I have seen any tree, neighborhood joining tree you made with the different strains of a fungus or virus or something, but you are doing a neighborhood joining method. What for? If you are doing the strain of the same thing, you should go for the parsimony, not for the neighborhood joining. Clear? So, depending upon your means a query, you have to choose what type of tree you want to generate, not that anything you will software, you will tell software will generate, any tree software will generate, but you cannot after that you cannot infer that what actually you would like to say. So, this bioinformatics and other things has many limitations also. There are many good thing, but limitation is that it can only predict, it cannot tell you, it cannot dictate you. Many times it gives erratic result, and most of the times this is due to the user who is using the software, and different subjects are interlinked. So, understanding is very difficult, and it may be possible to make the relationship or models, but in reality they may not work, because we are trying to take challenge with the mother nature who takes billions of years to evolve us, and we are trying to just sitting on the computer, we are trying to understand what is the relationship, what is the model, and all these things. So, we do not know who will tell the last answer, our beloved mother nature or the silicon chips. Thank you for your kind attention. Now, question, anything?
Now, the talk is open for any discussion. Stories. Of course, you will be handling one more practical. You can reserve those questions for practical. We are running out of time now. Right.
We can discuss. I am here only, so no problem. You can come anytime for your practical analysis also. If you need any help or anything, so you can come.
Bring some set of data for analysis. Whatever you told, character based, distance based, a set of data will give for all the groups. Let them do the analysis using these programs. Now we'll close this particular talk, and I request Dr. Annapurna to propose the formal.
Oh, thank you.
It's a great talk. Yeah.
Yeah. I think it was a very scintillating talk, I would say. And I'm sure most of you will be benefited by this talk. And many of your small doubts or queries, and probably you will now, in retrospect, if you think about this talk and think what you have been doing all along for your sequence analysis, you would know what were the common mistakes that you probably have done. And I hope you will, like you said, after this training program is over, we would at least expect this group of students not to repeat those mistakes. And we also hope that in future, since he is a part of IRA and all of you are students of IRA, if you have any doubt on this particular aspect, or maybe others also, I think Anirban, you are available for the student community.
Of course.
And I know he is an excellent teacher. This talk of almost 1.5 or 1 hour 45 minutes is an ample demonstration of that. And please take advantage of this. And like he says, no more confusions on this particular aspect at least. I would profusely want to thank you, Anirban. He is much younger to me, but he has really risen, and I feel very glad when I see such kind of students who have come up from here and are delivering such kind of talks where students are benefited. The whole effort of this training program is to see that the students of IRA are benefited on different aspects of genomics, and I really know that there is a huge confusion as far as genomics and the advanced tools and the advanced techniques are concerned. So our attempt is only to see that that kind of a confusion is removed, and for that please enjoy all the talks in this training program, and in future also please feel free to come and interact with all the faculty who will be now coming up one after the other that lectures have. Thank you very much. Thank you very much, Anirban.
When I was student, I faced these problems and I developed those things that how really to do, and I am very interested to tell you that how we have come up with those things, and you should do those things, because I also faced this problem because I was an alumni from this institute.
Yes.
So
in fact, we are all alumni of this institute, so that the reason for us to feel this is much more. And this is being telecasted, webcasted to others, and we have sent this link to many of the universities. So I hope the student community in total in India is being benefited by these kind of this kind of talks and other practicals. Thank you very much.
So thank you, Anirban, for a wonderful talk. And with that, we conclude the first lecture for the day.